This is an overview of an article entitled Common and Rare Coding Genetic Variation Underlying the Electrocardiographic PR Interval, published in the May 2018 issue of Circulation, Genomic and Precision Medicine. The electrocardiographic PR interval spans from the beginning of the P wave to the beginning of the QRS complex, as shown in the schematic here. The PR interval can be considered as a biomarker of atrioventricular conduction. To assess for genetic factors that contribute to clinical traits, genetic association studies are performed. The most commonly done association study has been the Genome-Wide Association Study, or GWAS, which assesses common DNA variants throughout the genome for strength of association with the phenotype of interest. These common variants are for the most part non-coding variants. More recently, exome chips that can directly interrogate coding variants in genes have become available. While the exome chips do cover some non-coding variants, they are very comprehensive with respect to coding variants. In the paper under discussion, the authors used exome chips to interrogate more than 220,000 high-quality single nucleotide polymorphisms, or SNPs, including 42,075 common variants, whether coding or non-coding. They performed their exome chip genotype association study in more than 92,000 individuals, most of whom were of European descent, with a smaller number with African ancestry. Here are the results of the exome chip analysis with the PR interval. In this graph, the x-axis is the position in the genome, split into different chromosomes, and the y-axis is the strength of association. The authors identified a total of 31 loci, meaning a stringent threshold for statistical significance. The most strongly associated SNP was a missense variant in SCN10A, which encodes a subunit of a voltage-gated sodium channel. Here, the coding variants in SCN10A that met either stringent or nominal statistical significance thresholds have been mapped with respect to their locations on the protein. This study identified a total of 21 new loci associated with the PR interval, out of a total of 31 associated loci. 8 of these loci are associated with atrial fibrillation as well. Besides SCN10A, rare variants in MYH6 and SCN5A were also found to be associated with the PR interval. Finally, PR-related variants were enriched in the heart morphogenesis pathway, suggesting that some of the loci might influence the PR interval through involvement in cardiac development.